You think darkness is scary? The ocean below 500 meters will change that opinion fast. Pitch black water, crushing pressure, temperatures near freezing. And in that void, things glow. Not gentle twinkling stars, not pleasant aquarium displays. These are organisms that evolved light as a weapon, a trap, a scream for help. 75% of deep-sea animals make their own light, according to research published in 2024 by the Monterey Bay Aquarium Research Institute. That same study analyzed 240 remotely operated vehicle dives and found bioluminescence everywhere. Not rare, not exceptional. Common. Standard equipment for surviving where the sun never shines. The midnight zone starts at 1,000 meters down. Here you find creatures with photophores covering their bodies, bacterial colonies living inside fish tissue, chemical reactions producing cold light that takes zero thermal energy. Some flash warnings, some mimic prey, some hunt with invisible wavelengths, and a few, a very specific few, combine adaptations so nightmarish they rewrite what you think predation means. We cataloged over 695 luminescent genera of marine animals in a study from March 2024, published in the journal Life, representing a 40% increase from the 1987 baseline. New species appear in deep water every expedition. The numbers keep climbing. Scientists now estimate over 1,000 genera produce light across all domains of life. The ocean midnight zone holds most of them. This script pulls from recent peer-reviewed research, including studies published between 2024 and 2025 in journals like Biological Reviews, eLife, Frontiers in Marine Science, and Deep Sea Research Part 1. Every claim here traces back to scientific observation. Every number comes from published data. Every creature exists with documentation. No speculation, no legend. Just what researchers found when they dropped cameras and nets into water deeper than most mountains are tall. This script is researched and factual, using sources from the Monterey Bay Aquarium Research Institute's 2024 quantitative bioluminescence analysis, examining over 30,000 individual animal observations across depths from surface to 4,000 meters, and from Cornell University's 2019 genomic study published in eLife analyzing bacterial symbionts in 7 species of deep-sea anglerfishes across 6 families. Collected from the Gulf of Mexico, these are not general marine biology textbooks. These are cutting-edge field studies with specimen counts, genetic sequencing, and controlled laboratory observations. Now back to the information. The most speciose fish group at those depths, according to Theodore Peach's 2009 taxonomic review. 9 of those 11 families evolved something extraordinary. A lure, the esca, a fleshy bulb dangling from a modified dorsal spine above the fish's head, containing living bacteria that glow blue-green 24 hours a day. 2 species of luminous bacteria colonize anglerfish bulbs based on genetic analysis from Lydia Baker and Tori Hendry's October 2019 study in eLife. They sequenced bacterial genomes from specimens collected in the Gulf of Mexico between depths of 0 to 1,500 meters. The bacteria genomes show 50% reduction compared to free living relatives. They lost genes for making amino acids, lost genes for breaking down nutrients except glucose, kept genes for building flagella to swim in water, kept some chemical sensors. The bacteria evolved to depend on the fish, but still survive outside it. Researchers found the same bacterial DNA floating in seawater samples at mesopelagic depths, where adult anglerfishes spend most time. The light organ develops later in life with a tiny pore opening to seawater. The bacteria colonize from the environment. Once inside, they reproduce constantly. The fish cannot turn them off. The bulb glows perpetually. To stop the light, the fish must physically block it with pigmented tissue. Some species in genera Cryptopsaras and Serradius evolved additional light organs called caruncles, pouch-like protuberances anterior to the dorsal fin, also packed with glowing bacteria. Males lack the lure entirely. No esca, no bioluminescent bacteria. They exist to find females and reproduce, then die. The size difference approaches 8 to 1 in some species. These transposons insert into functional genes and break them. The bacteria are actively losing genetic information, evolving toward obligate dependence, yet they persist in open water between hosts. How? Scientists found granules resembling polyhydroxybutyrate, or PHB, inside the bacteria, a carbon storage molecule. The bacteria may stockpile energy during their time inside the fish, then slowly burn it for decades in the cold, high-pressure water, waiting for the next larval anglerfish to mature and open its pore. At depths below 1,000 meters where anglerfishes hunt, that's the only light. No sunlight penetrates, just bioluminescence. The bacteria create a point source bright enough to attract copepods, small fish, anything curious or confused by light and total darkness. Then the anglerfish strikes. Jaw hinges built for extreme gape, stomach walls black to hide the glow of consumed prey. A mobile trap that never turns off. Everett Jones identified in 1971 that cigar sharks, now called cookie cutter sharks, caused mysterious crater wounds on marine mammals. Before that, researchers blamed lampreys, bacteria, parasites. The cookie cutter shark, Icystius brasiliensis, earned its current name from shark expert Stuart Springer, who originally called them demon whale biters. Ancient Samoan legend described skipjack tuna entering Palauali Bay, leaving behind pieces of flesh as sacrifice to the community chief Tautunu. They were describing cookie cutter bites centuries before science identified the culprit. Found in oxygen minimum zones at depths from 300 to 3,000 meters in tropical and temperate waters worldwide. Not actually a squid, not an octopus, something between. 8 arms like an octopus, but 2 retractile filaments between the first arm pairs on the dorsal side. The filaments have sensory hairs for detecting and capturing marine snow, detritus, fecal pellets, dead gelatinous zooplankton. The body reaches up to 30 centimeters total length, covered almost entirely in photophores, light-producing organs scattered across the mantle and arms, except the undersides of the webbed cape. The photophores emit blue light in flashes, ranging from fractions of a second to several minutes. The vampire squid can modulate intensity and size. Larger, more complex photophores cluster at the arm tips and fin bases. 2 white areas on the head, initially thought to be photophores, are actually photoreceptors, the largest eyes proportional to body size of any animal in the ocean. It curls arms up and outward, wrapping them back over the body, turning itself inside out. This exposes cirri, fleshy spines on the arm webbing that look dangerous but cause no harm. The heavily pigmented cape underside masks the body photophores. The glowing arm tips cluster together far above the animal, creating a false target. Attack the lights, not the body. The vampire squid ejects thick, glowing mucus from arm tips, not ink. Bioluminescent mucus containing photophores that continue emitting light after separation from the body. The cloud hangs suspended, confusing predators, while the squid uses its ear like fins to flap away. The mucus production was documented in laboratory observations and deep sea video from ROV encounters. Third mechanism documented in a December 2024 article from Scientific Inquirer. When the photophores flash in coordinated patterns combined with writhing arms and erratic movements, the display creates multiple simultaneous targets. A predator cannot track which glowing point is the actual animal. The bioluminescent fireworks multiply perceived threats. The vampire squid lives in the oxygen minimum zone, where dissolved oxygen drops below 0.5 milliliters per liter. Few predators tolerate those conditions. The squid evolved extremely efficient hemokinin, the copper-based blood protein that transports oxygen in cephalopods. Its metabolic rate runs incredibly slow. It can survive where most animals suffocate. Stomach contents analyzed from mature specimens contained fish bones and scales alongside gelatinous zooplankton. Young vampire squids drift as larvae. As they mature, they develop the cape and bioluminescence. The species existed essentially unchanged for 3 million years, according to phylogenetic analysis. A living fossil adapted perfectly to permanent darkness and low oxygen. 2 species, 2 wavelengths. Ideacanthus atlanticus, black dragonfish, found circumglobally in southern subtropical and temperate oceans between 25 degrees south and 60 degrees south, at depths down to 2,000 meters. And Malacosteus niger, stoplight loosejaw, found between 66 degrees north and 33 degrees south, except the Mediterranean, at depths below 500 meters, extending to 2,500 meters. Start with the dragonfish. Females grow to 40 centimeters, males to 5 centimeters. Extreme sexual dimorphism. Females are black with 6 stripes. Males are brown, lacking canine teeth, pelvic fins, and the female's chin barbel. The barbel is the lure, a glowing appendage hanging below the jaw tipped with a photophore. Flashing it on and off attracts prey in the darkness. The dragonfish also has photophores along its belly for counter-illumination, matching faint, downwelling light to erase its silhouette when viewed from below. A January 2025 article from How Stuff Works described it as the same aesthetic you would see behind the Terminator's sunglasses when angry. Most deep-sea organisms are insensitive to red wavelengths. Red objects appear black in the deep sea because red light from the surface doesn't penetrate past a few meters. The dragonfish evolved to both produce and see red light. Research published in Vision Research in 1999 by Douglas Partridge and colleagues found the dragonfish uses far-red emitting photophores and specialized rhodopsins in the retina to detect wavelengths other fish cannot see. This unique evolutionary trait appeared around 15 million years ago and had a single evolutionary origin within the Stomidae family, according to genetic phylogenetic studies. It can illuminate prey with an essentially invisible searchlight. The name stoplight comes from the red and green lights resembling traffic signals. Research from Douglas and colleagues published in Philosophical Transactions of the Royal Society B in 2000 found evidence the pigment comes from eating copepods. The copepods feed on bacteria containing chlorophyll derivatives. The fish obtains the pigment through diet, incorporates it into retinal tissue, and uses it to absorb red light around 670 nanometers, which then transfers energy to visual pigments tuned for 520 and 540 nanometers. No other vertebrate does this. No skin membrane connects the two sides of the lower jaw, just the hinge and one exposed muscle called the protractor hyoideus. This reduces water resistance, allowing the jaws to snap shut with incredible speed. The connection between head and body uses unossified vertebrae, so the cranium tilts back and jaws thrust forward for maximum gape. The gills expose directly to water, so the fish can breathe while slowly swallowing prey larger than its head. Fang-like teeth in front, many small barbed teeth behind, pharyngeal teeth directing food down the esophagus. This confused scientists. The fish has anatomical adaptations for eating large prey, but primarily eats zooplankton. The hypothesis goes like this. Large calanoid copepods are 3 orders of magnitude more abundant than fish or shrimp at those depths. The loose jaw uses red bioluminescence to search for copepods in a small illuminated area, invisible to everything else. It feeds constantly on abundant tiny prey. Occasionally, it encounters larger prey, and the extreme jaw mechanism handles it. The chlorophyll derivative required for red vision comes from the copepods themselves. The entire hunting strategy depends on eating the same organism that provides its visual superpower. 5.8 to 11.9% of upper jaw length versus 2.8 to 8.1% in females. Males also have larger eyes, potentially for detecting mates during sparse encounters in the deep sea, according to recent studies on sexual dimorphism published in 2025. Deep red coloration, 20 marginal tentacles and 1 hypertrophied tentacle, a long trailing appendage larger than all others thought to facilitate prey capture. Named after Sir Charles Wyville Thomson, chief scientist on the Challenger expedition. Depth range from several hundred meters to over 4,000 meters. When attacked, it produces an array of blue light flashes propagating at speeds of 5 to 50 centimeters per second in circular waves, according to research published by marine biologist Edith Witter and colleagues. The pattern resembles a rotating pinwheel of light wrapping around the bell circumference. This is not defense through confusion. This is recruitment. The jellyfish emits bioluminescent screams designed to attract larger predators. The attacking predator becomes visible to even bigger hunters drawn by the light show. The original attacker faces a new threat. Often, it retreats. The jellyfish escapes. Evidence from gut content analysis showed shrimp as a documented threat to Atolla wyvilli, according to a 1993 study by Moore. The alarm presumably targets predators of shrimp. Attract something big enough to scare off a shrimp, and you survive. The first footage of a giant squid in U.S. waters came from an expedition using Witter's E-jelly in the Gulf of Mexico. The device proved the alarm signal works as intended. Big predators respond to distress displays from smaller animals. The individual can control timing, duration, and intensity. The display is not random. It follows specific sequences and patterns. Researchers studying these patterns published findings in March 2024, confirming the light serves both defense and potentially communication functions. The dark red body coloration helps the jellyfish remain invisible in ambient conditions. Red appears black in the deep sea. But when it activates the blue flashes against that black canvas, the effect is a brilliant emergency beacon visible for tens of meters in every direction. It feeds on crustaceans and floating organic material, trapping prey with the extended tentacle and bringing it to the mouth at the bell center. The bioluminescence from captured prey could reveal the jellyfish location to predators, but the dark body pigmentation and light-emitting complexity of the jellyfish prevents that glow from being visible. It is why they live in extreme depths rather than near the surface. The adaptations only work in perpetual darkness. Large caudal fin with lower lobe nearly as large as upper lobe, which has a prominent ventral notch. Dark brown coloration becoming subtly lighter below, and around the gill region, a dark collar. That collar is the key. They produce vivid green bioluminescence, the strongest intrinsic luminescence known from any shark according to research published in Frontiers in Marine Science in April 2021 by Del Ruas and colleagues examining photophore distribution and enzymatic diversity. The light persists for 3 hours after the shark is removed from water. Individual photophores are set around the denticles and are small enough they cannot be discerned by naked eye. This suggests they evolved to fool animals with high visual acuity at close distances. The collar is darkly pigmented and contains very few photophores. It does not luminesce. When the rest of the shark glows to match background light, the collar remains dark. From below, it resembles a small fish silhouette. The shark breaks up its own silhouette, then presents a false target where the collar sits. Larger predators looking upward see what appears to be a small fish swimming alone. They approach to attack the apparent prey. That brings them within range of the cookie cutter shark's suctorial lips and specialized pharynx. The shark attaches using powerful suction. Then it uses its bandsaw-like set of lower teeth arranged in a single row of triangular, erect interconnected plates to gouge out a neat circular plug of flesh. Cookie cutter wounds have been documented on whales, dolphins, seals, large fish, submarines, undersea cables, and human bodies. A drowning victim recovered off Oahu, Hawaii in July 1992 had 2 cookie cutter bites to the lower back. Another human body, a suicide victim, also showed cookie cutter damage according to reports from ABC News. It rises from depths around 2,000 to 3,000 meters up to the surface at night, then descends before dawn according to research published in 2012 by Wenzel and Lopez Suarez. Cookie cutters traveling in schools create the deception of a school of small fish when their dark collars are visible against glowing bodies. This multiplies the lure effectiveness. They are facultative ectoparasites. They feed on tissue without killing the host animal. A dragonfish photographed with cookie cutter wounds continues living. A whale with crater scars survives. The shark takes enough to feed but leaves the victim alive. It can hover motionless, waiting for prey to investigate the false target. Energy-efficient predation in a food-limited environment. Genetic studies of the light-producing enzymes found multiple luciferase variants, suggesting the bioluminescent system evolved sophisticated regulatory control. Every detail comes from peer-reviewed scientific literature, including studies from the Monterey Bay Aquarium Research Institute, analyzing quantitative bioluminescence across depths, and from the Frontiers in Marine Science, 2021 examination of cookie cutter shark photophore distribution using histological analysis and transcriptome sequencing of light organ tissue. These are not Wikipedia summaries. These are primary research papers with specimen collection data, genetic sequencing results, and controlled experimental observations. Now back to the information. Found in mesopelagic and bathypelagic zones from 200 meters down to 5,000 meters in tropical and temperate waters worldwide. The species dominates submarine calderas like the Carosa Hole, which has the highest Chaliotis density known anywhere in the world. The first vertebra behind the head acts as a shock absorber. The viperfish is thought to impale victims by swimming at them at high speeds. The teeth lock the prey in place while the fish manipulates it toward the throat. Flashing the light on and off creates a lure effect, drawing smaller fish within striking range. Additional photophores line the sides of the body. These serve counter-illumination, making the bottom of the fish appear to blend with extremely faint light filtering from above when viewed by predators lurking below. This dual use of bioluminescence, hunting, and camouflage appears across multiple deep-sea predator lineages. Viperfish feed primarily on crustaceans and small fish. Stomach contents from captured specimens contain lanternfishes, bristlemouths, copepods, and krill, according to research analyzing feeding ecology. They perform diel vertical migration. During daytime, they inhabit depths down to 5,000 meters. At night, they travel up to shallower waters less than 2,000 meters, where food is more abundant. They can survive temperatures from 4 to 14.5 degrees Celsius, tolerating daily temperature changes up to 7 degrees Celsius, according to research on vertical habitat and temperature tolerance. They are known to be caught in nets as bycatch, though their deep habitat keeps human interaction minimal. The bacterial presence suggests some viperfish bioluminescence is bacteriogenic rather than autogenic, meaning produced by symbiotic organisms rather than the fish's own chemistry. They reproduce through spawning. Studies on related dragonfish species showed males spawn sperm continuously, while females display asynchronous oocyte development and batch spawning. One Gulf of Mexico study found a skewed 1-to-2 sex ratio favoring females in over 70 Chaliotis sloanii specimens collected. In tropical waters, viperfish tend to stay in deep layers without much migration. In temperate waters, they migrate more actively and interact with epipelagic predators. They have been recorded in Italian waters off the western Mediterranean basin, the Adriatic Sea, Greek waters of the Aegean Sea, and Turkish waters of the Levant Sea. Not to be confused with freshwater hatchetfishes, which belong to a completely different family. The scientific name Sternoptychini derives from ancient Greek sternon, meaning breast, and pitix, meaning fold or crease, referring to the thorax shape. Found in tropical, subtropical, and temperate waters of the Atlantic, Pacific, and Indian Oceans, ranging in size from Polyipnis dani at 2.8 centimeters to the giant hatchetfish Argyropelicus gigas at 12 centimeters. Sternoptyx and Argyropelicus are decidedly angular when viewed from the side. The mouth is located at the tip of the snout, directed almost straight downward. Scales are silvery delicate and easily abraded. In some species, like the highlight hatchetfish, Sternoptex pseudobscura, large sections at the base of the anal and caudal fins are transparent. The eyes are large, sometimes tube-shaped, capable of collecting the faintest light and focusing on objects both close and far. They are directed somewhat upward, most conspicuously in genus Argyropellicus. This allows them to discern silhouettes of prey moving overhead against slightly brighter upper waters. They hunt by looking up. Hatchetfish have bioluminescent photophores distributed along their bellies in precise patterns. These produce blue light. They use this for counter-illumination, matching the faint downwelling light to eliminate their shadow when viewed from below. But the sophistication goes beyond simple on-off switches. Research published in 2021 in the Proceedings of the Royal Society, B found evidence of eye-facing photophores. A reference photophore, directed toward the eye with intensity, correlated to ventral photophore output. The fish can monitor its own light emission and adjust it in real time to match ambient conditions. In shallow parts of their range where solar light dominates, hatchetfish may redirect sideways sunlight down through photophores, assisting counter-illumination. In deeper parts, where they face searchlight hunting from other bioluminescent predators, dumping light downward throws predators off trail. The scales use photonic structures that control light at the nanoscale level according to physicist research examining the biological materials. Hatchetfish typically occur a few hundred meters below surface, but their entire depth range spans 50 to 1,500 meters deep. They perform diel vertical migration, in some species rising at night to feed on plankton and small fish, then returning to darkness by day. Most researchers agree they have short lifespans no longer than a year. Reproductive habits remain mysterious, though juveniles look much different than adults. The photophore patterns differ slightly between species. Many researchers believe the patterns play a role in courtship, though very little is known about mating. The ability to modulate bioluminescence intensity and distribution in real time, depending on ambient light levels, represents remarkable evolutionary precision. The hatchetfish essentially wears adaptive camouflage that recalibrates constantly as it moves through different light zones. 3 genera exist in the subfamily: Argyropellicus with 7 species, Sternoptex and Polyipnus with additional species, bringing the total to around 40 species. They are found in most temperate waters worldwide, typically at depths from 600 feet to 4,500 feet. A specimen from the Gulf of Mexico, examined by researchers, showed the half-naked hatchetfish, Argyropellicus hemigymnus, has 3 forms of camouflage working simultaneously. A compressed body with silvery mirror-like flanks, reflecting light from angles, making it invisible. Bioluminescent photophores on the underside, for counter-illumination matching downwelling light. And transparency in the tail region that wiggles, making it appear partially invisible. Triple redundancy in a fish smaller than a human hand. In the ocean specifically, it arose multiple times within different lineages. The chemical pathways vary. Some organisms use bacterial symbionts. Others produce their own luciferin and luciferase. Some use cholinterazine. Others use different molecules entirely. No photosynthesis below 100 meters. The food web depends entirely on marine snow drifting down from surface waters and on predation. In this environment, bioluminescence became standard, not optional, not rare, required equipment. Eyes evolved during that same period. The ability to produce light and the ability to see light co-evolved in the ancient oceans. Predators developed better vision. Prey developed better camouflage using light. An evolutionary arms race playing out in darkness for 500 million years. Medical imaging uses bioluminescent proteins for cancer detection and cell culture research. Green fluorescent protein from the crystal jellyfish, Equirria victoria, revolutionized cellular biology. Researchers can tag specific proteins and watch them move through living cells in real time. The discovery won the Nobel Prize in Chemistry in 2008. Avoid predators, find mates, communicate warnings. Every flash costs energy. Every photophore requires maintenance. The organisms would not invest resources unless the return justified the cost. And across 75% of deep-sea species, the return clearly justifies it. The midnight zone is not dark. It is filled with cold, calculated light. November 2024, researchers described Bathydevius caudictilis, the mystery mollusk, a new species of nudibranch from the midnight zone. It swims with a large gelatinous hood and paddle-like tail. When threatened, it lights up with brilliant bioluminescence. It can detach a steadily glowing finger-like projection from the tail as a decoy. Over 20 years of research from the Monterey Bay Aquarium Research Institute went into describing the single species. It is one of more than 250 deep-sea species documented by MBARI technology previously unknown to science. March 2025, Research published in FIU News revealed deep sea shrimp evolved enhanced vision using multiple opsin proteins to see different colors of bioluminescent light, including environmental blue light, and possibly differentiating between their own glow and bioluminescence from other species. The shrimp genus Oplophoroidea migrates vast distances up and down the water column. Those migrating into shallower waters have greater variety of light-detecting proteins because they encounter more complex light environments. Vision, adapted to a bioluminescent world, operates on entirely different principles than surface vision. Others use photophores embedded in tissue. Some do both. The diversity of mechanisms within a single taxonomic group suggests intense evolutionary pressure to develop light production. It is not enough to have light. You need the right kind of light used the right way at the right time, or you die. Black dragonfish and stoplight loosejaw use invisible wavelengths. Atala jellyfish use recruitment signals. Cookie-cutter sharks use deceptive camouflage. Viperfish use fishing rod lures and counter-illumination. Hatchetfish use real-time adaptive camouflage. Each evolved independently. Each solved the problem of survival in darkness using light. The nightmare is the sophistication, a bacterial genome losing genes while maintaining environmental persistence. A visual system that steals chlorophyll derivatives from food to see invisible wavelengths. A jellyfish that screams for help using light that travels through water as circular waves timed to specific propagation speeds. A shark that mimics prey using the absence of light surrounded by light bright enough to persist 3 hours after death. These are not simple organisms responding to stimuli. These are complex systems executing strategies that took millions of years to refine. More than 70% of the planet's surface is ocean. The average ocean depth is 3,700 meters. Most of that volume sits in perpetual darkness. And in that darkness, most things glow. Genetic sequencing that can analyze bacterial symbionts from a single specimen, computer modeling that can simulate light propagation through water and test counter-illumination effectiveness, submersibles that can collect live specimens and keep them alive during decompression. Each technological advance reveals layers of complexity previously invisible. The burglar alarm hypothesis for Atala jellyfish came from direct observation of attack responses filmed in situ. The chlorophyll derivative visual system in stoplight loosejaw came from retinal tissue analysis using spectroscopy and microscopy. The bacterial genome reduction in anglerfish symbionts came from DNA sequencing comparing hundreds of specimens. The cookie-cutter shark's differential blending strategy came from mathematical modeling of light fields combined with behavioral observations. None of these discoveries happened by accident. They required decades of systematic research with peer review and replication. Over 6,000 luminescent species from approximately 1,000 genera. That number will increase. Every major oceanographic expedition finds new species. The deep sea contains more biodiversity than tropical rainforests, according to some estimates. Most of it remains undocumented. The anglerfish cannot hunt without bacterial symbionts. It must acquire from the environment and then can never turn off. The vampire squid cannot escape predators in low oxygen zones without bioluminescent decoys and false targets. The dragonfish cannot see prey without producing light wavelengths that should not exist in the deep sea and stealing plant pigments to detect them. The Atala jellyfish cannot survive without recruiting its enemies' enemies using precisely timed light displays. The cookie-cutter shark cannot feed without creating a silhouette of prey from the absence of its own bioluminescence. The viperfish cannot attract food without a lure that might also attract predators. The hatchetfish cannot hide without real-time light matching that requires eye-facing photophores for calibration. Remove the bioluminescence and these animals cannot exist. The adaptations are not optional modifications. They are core survival requirements. The light is not decoration. It is weaponry, camouflage, communication, and sensory system combined into organs smaller than sesame seeds, producing cold light through chemistry that generates zero heat and operates at pressures that would crush human submarines. The ocean midnight zone is not empty darkness. It is a battlefield illuminated by living organisms where every flash means something. Prey detected, mate located, predator warned, help requested, threat displayed. The creatures that evolved there develop solutions that seem impossible. Bacteria that survive decades in crushing pressure while genome reduction continues. Fish that see colors nothing else can see using molecules borrowed from food. Sharks that glow brighter than any other shark species to hide by becoming invisible through selective darkness. Jellyfish that turn predation into recruitment. Research from Mbari, published in November 2022, analyzing over 30,000 individual animal observations, found the proportion of glowing to non-glowing animals stays relatively constant from the surface all the way down to 4,000 meters. Bioluminescence is not more common in deeper water. It is equally common at all depths below 200 meters. The midnight zone does not have a monopoly on bioluminescence. It just has the only place where bioluminescence becomes absolutely necessary rather than merely advantageous. Instead of adjusting light intensity like other animals, sharks emit constant light output and move up and down in the water column to remain cryptic at the depth where their luminescence matches ambient light. They depth match rather than intensity match. This explains why cookie-cutter sharks perform such extreme vertical migrations. They are searching for the isoluminance zone where their particular light output makes them invisible. The adaptations go deeper than individual species. Entire ecosystems structure around bioluminescence. Lanternfish, small fish with rows of photophores, perform the largest daily migration of vertebrates on Earth. Billions of individuals rising from depth at night to feed near surface, then descending before dawn. Their bioluminescence serves counter-illumination, but also mate attraction and potentially predator confusion. They form the base of many deep seafood webs. Everything from squid to whales depends on them, and they depend on their photophores to survive the vertical journey. But the energy cost is lower than other adaptations like active swimming or building heavy armor. In an environment where food is scarce, bioluminescence offers the best return on investment. A small photophore producing brief flashes costs less than constant movement or physical defense structures. It produces solutions that work well enough to survive and reproduce. These organisms work far better than well enough. They dominate their environment. The anglerfish is the most specious fish in the bathypelagic zone. The cookie-cutter shark parasitizes prey 10 times its size. The vampire squid survives in oxygen levels that kill most animals. The hatchetfish has triple redundant camouflage. These are not struggling survivors. These are apex solutions to specific problems. Most of what we know about these animals we learned in the last 50 years. Most of what we will know has not been discovered yet. And filled with organisms that produce their own light through chemistry, we barely understand using it for purposes we are still documenting in research published last month and next month and every month as long as we keep looking. Each one represents millions of years of evolutionary refinement, producing an organism so specifically adapted to its environment that removing a single feature would cause extinction. You cannot have an anglerfish without bacterial symbionts. You cannot have a stoplight loosejaw without chlorophyll derivatives. You cannot have a cookie-cutter shark without the dark collar. The adaptations are irreducible. They work as complete systems, or not at all. Soil cores from hydrothermal vent sites show bioluminescent bacteria have existed for billions of years. The chemistry predates multicellular life. When the first animals with eyes evolved, bioluminescence was already there. The deep sea arms, race between light production and light detection, started before the Cambrian explosion, and continues today in water so deep and dark, humans needed advanced technology just to know it exists. That is the nightmare. Not that they glow, that they had to.